So I am really happy to be here to talk with all of you, because I think Simon's has really been not just a thought leader, but really kind of an operational tour de force in moving forward the field of autism research in a variety of different areas. And I think that hopefully some of the areas I touch on today, you'll really sort of appreciate how I think the Simons Foundation has either directly or indirectly really been helpful in kind of helping us think about the initiatives that we are moving forward with. Quickly, just my financial disclosures up here, but I like to actually really emphasize kind of more of a personal, professional disclosure, which is that while I am going to talk about my research and I am a researcher, I actually fundamentally at heart am also a clinician. I'm a child neurologist and I take care of and diagnose patients with autism, neurodevelopmental disorders, and largely infants and children who are newly diagnosed with genetic etiologies for their autism or neurodevelopmental disorders. And what I see in clinic is this: parents come to me with the number one question of, why, not simply why has this happened, but what's next? What's the right treatment? And they often will seek guidance from, our esteemed colleagues of Google and Wikipedia and all the other internet, outlets there, especially in Los Angeles, there are many. And this is what I see, is that people are just taking treatment after treatment after treatment, and they're being offered cures, and they're being offered, dramatic results. And often by the time they come to me, they've already tried all of these various, things, some of which could be dangerous, some of which may not be dangerous, but clearly are not evidence-based. And so a lot of the research that I'll talk about today, and in general, I'd say like our research program at UCLA and in my lab, is really driven by this goal of being as rigorous and evidence-based in our design of studies that will ultimately enhance treatments, right? So ultimately we want treatments, but we can't say, oh, I want a treatment, I'm gonna go out there and just try this treatment on a bunch of kids and see if that works, right? We need rigorous clinical trials that are designed in a way that allow us to understand why a drug works, whether it truly works, how it works, and which populations might it work best in. And that's a lot harder than we might think, especially in the space of autism. And so that's really, this kind of clinical reality is really what drives the research. And I'm lucky to be able to do the research in CART, as Paul mentioned. CART is a multidisciplinary autism center at UCLA. We're lucky to have been fairly well funded. We just received our third Autism Center of Excellence, and I'll talk a little bit about that. But what I love about what we do at CART is we integrate neuroscience and basic research with clinical practice and intervention. And so a lot of the work we do in intervention trials even is really inspired by what we're learning about brain development and function in autism. So in that setting, what I'm going to do today, and succinctness is not my strong suit, but I have lots of clocks all over, so I'm going to stick to my time. I'm going to start by laying kind of a very broad framework about what I think are kind of challenges and gaps in the field of clinical trials and treatments in autism. And then I'm going to talk about how we might sort of think about filling that gap with our research in biomarkers. And I'm going to focus on EEG biomarkers, which is the area that I have expertise in and what my lab really focuses on. And then I'm going to give you kind of a data blitz of some of the studies we're doing in EEG biomarkers, really to give you kind of a broad perspective on the field and kind of what we're doing in each of these areas. Again, each of those little blitzes could be a talk in itself, but I thought it would be nice to give you kind of a good overview rather than focus on one area. And then I'll end with sort of some thoughts, call to action, and next steps. Okay, so I have to give you my obligatory what is autism slide. You all know this, but what I want to emphasize about the fact that autism is diagnosed by impairments in social communication skills and the presence of repetitive behaviors and restricted interests is a few things. So one is that it's a neurodevelopmental disorder. We know that or condition. We know that symptoms emerge early in life. Sometimes symptoms might emerge before we make a formal diagnosis, and we leverage that in some of our high-risk studies. But really the key is that the diagnosis is based on a list of behaviors, right, that we read out of our DSM Diagnostic Statistics Manual or by features that we learn about through caregivers and parents and other people. But we know, based on many advances in genetics, that there's many different biological and genetic etiologies that can converge on this list of symptoms, if you will. Having said that, because there are many etiologies, we also appreciate that there's quite a bit of phenotypic heterogeneity in autism, right? The classic sort of Lorna Wing statement of if you've met one child with autism, you've met one child with autism is actually, somewhat true. Now, some of the common threads based on diagnosis are there, but there's so many individual differences. And that makes studying autism as a unified construct a very hard thing, and perhaps not something we should be doing. Maybe we should be thinking more about subgroups. And I just put this out there because despite this appreciation of heterogeneity, what happens in treatment, and this is largely based on sort of what we have evidence on and also what's available in the community, so partly practical, is that we think about symptoms, right? In our case, the list of behaviors. And we have an array of behavioral interventions that we use to treat those symptoms. And frankly, the behavioral interventions I recommend to patients are not necessarily based on what we know might be evidence-based. It's based on what the insurance will cover and where they live and what's available. And sometimes they can be very effective, but we don't always know ahead of time. And when we're using and recommending those treatments, we're looking at outcomes that, again, are kind of basic behavioral outcomes based on, again, the symptoms the child has. So this is kind of, I don't want to say as extreme as a one-size-fits-all approach, but it's a pretty general approach to recommending treatments when we know that there's so much biological heterogeneity and phenotypic heterogeneity. What we'd like to move to, and this is kind of the buzzword in the field, biomarkers is a big buzzword, the other big buzz phrase is precision medicine, right? Is that we'd like to move to a place where we can start thinking about targeted treatments, right, that are targeting ideally the etiology or maybe some of the changes in brain development that are occurring based on the etiology, and also maybe treatments that are targeting specific symptoms that are more specific to whatever that etiology might be, right? So we start with thinking about etiology, we think about what's happening in brain development, we might measure some targeted symptoms and have treatments that then target those things. Now, unfortunately, even when we've had situations where we have more targeted treatments, and I'll give one quick example on this in a minute, our trials still seem to, quote unquote, fail. What does fail mean? It means that they don't show us an improvement in the primary outcome measure of interest. And the example I'll use, and I don't mean for this to be an example of a failure because I think we've learned more, so much more than we maybe even expected to in the Fragile X story, is in Fragile X, where there have been multiple potential targeted therapeutics, the two big ones being a GABA agonist, arbaclofen, and the other being an mGluR antagonist, where there was tremendous hope based on preclinical models that these agents would actually reverse social impairments or difficulties in patients, because they did in the preclinical models. And then there were even studies showing safety, and then phase 2 and then phase 3 trials did not show any improvements in the primary outcome measures. And you can see, though, this is a really beautiful figure from a recent review that Paul was a part of, amongst others, led by Liz Berry-Kravis, where the outcome measures in all these trials, and they actually reviewed all the drugs that have been tried in Fragile X. There's a range of outcome measures that were used here, and none of them really showed a primary benefit, if you will. Now, there were some secondary outcome measures that did show some change. But all in all, as a body of work, while we learned a lot, and we're going to get into that in the talk, we did not find what we maybe were hoping for, which was this targeted treatment really showed a phenomenal improvement in a specific outcome measure. And I'll remind those of you in the room who aren't familiar with clinical trials, it's a very simple cartoon. What are we doing in these trials? We're taking a group of patients, we're randomly splitting them into two. A group gets a placebo, they're in a control group, and the other group gets the intervention, and we're basically measuring that outcome, whatever that outcome may be. Okay, so why in that setting, we need to start thinking about why are these clinical trials failing? And again, we're putting this in the context of how do we improve our chances for success? Now, I sort of break down the sort of clinical trial space into these sort of three areas. So one, it could be the drug, right? The drug might be missing the target. We actually may not. We may have a drug that seems to change Fragile X-like behaviors in mice, but it's not changing things in humans because it's not actually the right drug for humans. So that's possible. And I don't want to specifically focus on Fragile X. We can sort of broaden this to other neurodevelopmental disorders or autism as a whole. So a clinical trial might struggle because the drug doesn't hit the target. It also might be that our patient selection is flawed for some reason. So the one big one that we think about in neurodevelopmental disorders is our patient population might be too old. Many times in clinical trials, we have to study the drug in adults first and then go to children because of safety concerns. But adults are not just big children, just like children are not just little adults, particularly from a neurodevelopmental standpoint. But we're studying these drugs in adults. So that might be a problem when we're looking for outcome measures that may not change so much in adults. It might also be just that our patients are too heterogeneous. We're taking such a mixed bag of folks that some might respond, some might not respond, but we lose the effect of that big outcome difference because we have such a big variable group of patients. And then it also might be outcome measures, and this is what we'll kind of focus quite a bit on when we talk about biomarkers. But what are the outcome measures that we're really using? And they're behavioral outcome measures or clinical outcome measures. One concern is that our outcome measures might be a bit too subjective, meaning we're asking either parents or caregivers or even clinicians to report change. Now that, of course, the most important thing is we want parents and caregivers to see change, but there are sometimes inherent biases in filling out a survey twice over a trial, or even filling out a survey once and knowing what your child maybe didn't do quite right, and even teaching your child to do those things because you realize, oh, those are things that I think my child should be able to do, which leads to, what we consider possibly a placebo effect, or just, as we think of it, sort of test-retest issues. The outcome measures may be insensitive to change. So if we're looking at things like cognition, does cognition or an IQ change in 8 weeks with a treatment? Maybe not. So that's another challenge with our outcome measures. These are timers, sorry. And then I would say that the other sort of big issue with our outcome measures is they may fail to capture meaningful variability in our patients, especially those who have significant areas of challenge or difficulty, such as kids with very low IQs. Many of our kids with genetic syndromes have very low IQs, and when we're using a measure like a standardized IQ measure, when all the kids are falling at the floor, it's very hard to sort of see meaningful change or even variability in those measures. So our outcome measures are definitely challenged. And this is where we can start thinking about biomarkers as one way to fill the gap. And again, what I'm going to talk to you about here is sort of an introduction to this field and show you some data, but I'm not pretending to say that we've solved the problem with biomarkers and now we're going to have perfect trials. But I think this is where I'm laying the sort of foundation for where the challenges are so we can think about why biomarkers could be helpful. So biomarker is a characteristic that's objectively measured and evaluated as an indication of a biological process. It could be a normal biological process, a pathogenic process, or any process that reflects a pharmacological response to a therapy. So we use biomarkers in medicine all the time, and I won't bore you with all the examples, but you can think of them. Blood pressure for hypertension, blood glucose for diabetes. We have really robust biomarkers that we use in medicine. Now, as you can imagine, thinking about biomarkers in neurodevelopmental disorders is much more challenging. And hopefully you guys will get this slide up because this is an important slide. So you're all looking at each side. So how can biomarkers help us with this rubric of what I talked about in terms of challenges, right? So if you guys can see up there, and sorry if you can't, I'll flip back to the slide when we get up back on the big screen so you can see this. We think about drug, patients, and outcome measures. So biomarkers can help us from the standpoint of the issue with is the drug hitting the target. Biomarkers can help identify whether the drug has hit the target. They can be measures of target engagement. And I'll show you a possible example of that later. With regards to patients, they might be able to help us stratify patients a bit more, right, based on their underlying biology. And then finally, from the standpoint of outcome measures, biomarkers might serve as more objective outcome measures. Now, when I put this up, sometimes people say, but why do you care if, some EEG measure changes? What you really want to know is there clinical improvement? That's what we're hoping to change. And that is fundamentally true, and I agree with that. But it might be that in a short trial, that fundamental clinical measure has not yet changed, but that a biomarker might be a good intermediate outcome measure. It suggests that, hey, this drug is hitting the target and we're seeing some movement. Now let's keep going with this trial or this arm of treatments that then we can see the clinical outcomes that we would like. So for that reason, biomarkers, oh God, this is on a timer. For that reason, biomarkers can be quite helpful. So again, thinking about it this way, these are really the goals and when we think about biomarker studies. So specifically, not even specific to autism, but in general, but the first line definitely relevant to autism, what else do we need in a biomarker? So you talked about conceptually how they're helpful. Well, we'd like them to be grounded in mechanism, and we'll talk about that next, what that might mean. We'd like them to be developmentally informed. These need to be sensitive to development, given that we're thinking about treating a neurodevelopmental condition. And then there's really the practical stuff. They need to be practical. We need to be able to actually assay these things in patients. So, something like taking samples from brain tissue, probably not so practical for a clinical trial, but can we assay brain function in other ways? It needs to be scalable, something that doesn't have to be necessarily done in a research setting or a clinical setting. It would be nice if it was able to be done in the community. Translational potential is one I won't talk about a lot today, but wouldn't it be great to have a biomarker that actually can also be assayed in some of our preclinical models to see if when we're doing drug discovery, we can see changes in those things in preclinical models. And then this last one is one that we talk about a lot, and I put it in question marks. I don't know how strongly I believe in this, is that do we want the biomarker to actually relate to clinical symptoms? And I think it's a more sort of philosophical, deeper question that we can talk about later. I think if the biomarker is really related to mechanism, it may not relate to sort of some downstream clinical measure, and that may be okay. But we do have to understand what that mechanism is that's being measured. So it's complicated. Pharmaceutical companies like our biomarkers to relate to clinical symptoms, so we look quite a bit when we do our studies. Okay, so when we think about mechanisms, what kind of biomarkers might we want to use? This is where genetics gives us really nice insights into sort of the mechanisms that we want to be measuring. And this is one review that also really, I'm presenting kind of an oversimplified view of what we've learned from autism genetics, largely by many of the studies done here funded by Simons. But we know that based on genetic etiologies of ASD and the genes, the putative genes that are involved, that the effect on brain development is really fundamental. And we see differences in things like cortical migration and growth and in functional connectivity, in the way that synapses form and function and the way circuits develop. And also in kind of this very general, as we think of it, balance of excitatory and inhibitory circuits. We also know that these changes occur very, very early in brain development. This was a really nice paper that came out a couple of years ago, and they really looked at expression of these different gene groups, and we don't have to worry about those details, but they really are genes that are affecting these processes that I just talked about. They looked at where they were expressed in the brain and when in brain development. And you'll appreciate up here, this is early fetal to late fetal development. Most of these genes are really coming online very early in development. And so this has kind of led to this, I love this term that Dan Geschwind and Pat Levitt coined, this idea that autism is a developmental disconnection, in that these connections are just not forming quite in the way that they might in typical development from really early in brain development. We know from infant imaging studies, and this is work done by the Infant Brain Imaging Study, IBIS, which has studied high-risk infants, and we'll talk about high-risk infants in a minute, where they actually performed MRI, really beautiful MRI studies in a huge comprehensive consortium of babies at risk. And what they found really corroborated what we talked about with the genetics. There's differences in these babies in the first year of life in both inter and intra-hemispheric connectivity, in the morphology of the corpus callosum, which is a really important kind of connection between the right and left brain, as well as integrity and development of white matter fiber tracts. And this was done, again, through a big consortium of about 300 babies and imaging. So we know that these basic processes of sort of circuit development are affected in ASD. So how do we then use that to think about a meaningful tool to study biomarkers? And this is where I became very interested in EEG, and it's not just me, there's many people around the country doing work in EEG, and I would say that, no pun intended, some of this work is still in its infancy because of the fact that there's a lot of methodological challenges with EEG, which we'll talk about. But what is EEG? So it stands for electroencephalography. Many of you here may know quite a bit about EEG. I'm going to give you some pretty methods-heavy slides and then some kind of overview slides to sort of fit everyone's probably level of interest. But what we do with EEG is we're at the scalp, we're measuring the firing and functioning of neurons. So what we know in the brain is that when two neurons fire and communicate via the synapse, there's a tiny electrical signal that's actually generated. That you cannot measure at the scalp. But when thousands of neurons fire and sync, you can actually measure a really nice signal at the scalp with a very expensive amplifier that we have to buy to amplify the signal so we can see it. But needless to say, we can measure this synchrony. So it's actually really beautiful. We can actually just measure the robustness of synchronous firing of neurons at the scalp. So we can leverage that. We can take that EEG signal and do quite a few things with it. First, we can take that signal and break it down into its component parts. And each of these are different frequency bands that we name based on their frequency, from delta all the way to gamma. These different oscillations actually have very specific neurobiological and even functional significance. And they also have very nice relationships to typical development, which we'll talk about in a minute. But we can quantify this. So we can ask how much of this signal is attributed to by theta oscillations versus alpha oscillations. And we can calculate what we call power based on that, oscillatory power. And oscillatory power is really the marker of neural synchrony. It's the marker of the synchronous firing of these neurons. And so indirectly is really a marker of and a measure of brain connectivity. We can also, though, more robustly measure brain connectivity, and there's multiple ways to do this, using things like EEG coherence, synchronization, likelihood. There's lots of different ways. We use phase coherence, and we'll talk about that in a minute, but we can actually map cortical connectivity based on this raw EEG. I think my earpiece has fallen off, hasn't it? Oh, I was waiting for him to come. Just causing all kinds of problems. Do you mind if I keep talking while you do this?
Yep.
Okay, so the clock is saying 39 minutes. So what we also know about EEG is that the changes in EEG follow a developmental time course. And so they can really shed nice light on both typical and atypical development. So generally we know that sort of slow oscillations like delta and theta kind of are very prominent early in development. And then, were you guys able to hear me before this came? Okay, good. I was like, oh, wow, must have been quiet. Okay, so slow oscillations are very prominent early in development, and then over early infancy, those slow oscillations kind of attenuate, and the faster oscillations, which really more represent kind of cortical binding of local circuits, especially around tasks, those become more prominent. Alpha in particular is something we've been very interested in because it's incredibly sensitive to sort of basic subcortical to cortical connections, and is also very sensitive to typical development. So we can measure things like the peak at which the alpha frequency occurs, and alpha oscillations, and coherence in the alpha band, and it relates very nicely to typical development. And here's just an example of something like that. This is like a power spectral density plot. So this is power, so the amount of each kind of frequency, and this is all the frequencies in one EEG recording in a baby over time. And you can see that early on, there's always much more. There's more prominence of those slow oscillations. But over time, this is at 9 months, we start seeing this peak in alpha, which really becomes prominent by 12 to 15 months. And then over time, that peak becomes a little bit later, so that all of us have a peak alpha that's a little bit slower or later than what we see in infancy, early infancy. And we can measure these kinds of things as a marker of robust brain development, if you will. So we talked about kind of the fact that EEG is definitely grounded in neurobiological mechanisms and is developmentally informed. But as you may imagine or know, EEG also has a lot of practical value. It's much more practical than putting a child in an MRI and requiring them somehow to be still or swaddling them like crazy so that they're still. It's also scalable, and I'll show you an example of that at the end, where we might do our high-density EEG recording in the lab, but we can also collect EEG data from low-density arrays, either from their clinical hospital recordings if they're coming in for a clinical EEG, or just from lower-density CAPs and other kinds of arrays that are being developed quite a bit now because of the interest in this area. So I tell you all this, and I want to be clear that this is not a bright idea that we had and no one else has already thought of this. EEG has been around for quite a bit, and people have been trying to use EEG to study autism quite extensively. Thus far, the research has largely been focused on focusing on group differences and trying to identify are there EEG patterns that seem to be more diagnostic or consistent with an ASD diagnosis. And I showed this slide really just for effect. These are some reviews that have just looked at all the EEG studies in autism. This was a recent one by Mayada Elsaba and her colleagues, and they just looked at studies in connectivity in autism. And what they found, blue is under connectivity, gray is inconclusive, red is over connectivity. So we're all over the map here. And this is another review that was in Journal of Neurodevelopmental Disorders. You're not expected to be able to read this. Again, it's for effect. And they looked at every possible frequency band in every possible region and what's been found. And the results are everywhere. And so this is a challenge. And the question is, why is this? We have all these one-off studies that are not replicated. What is the reason for this? And there are several reasons, but I think that these are some of the really big ones, and this is what we try to sort of think about as we're designing biomarker studies and really thinking about what the key areas of interest are when we're using EEG to study autism. So one is that many of these studies use, and again, this is across the field, it's not a criticism so much as sort of a observation, is that we have small sample sizes that are really assaying sort of snippets of the entire spectrum. And so then if someone tries to replicate with another sample that might not be entirely representative, we may not replicate those findings. Also, I would say the majority of the studies in EEG have been focused on a very small subset of the autism spectrum, largely older individuals who have high IQs, because they're easier to study sometimes. And so we don't end up actually reflecting what we think might be occurring across the spectrum, or we're not capturing what we really want to know when we talked about, earlier with the biomarkers piece, which is how might we use EEG to stratify kids? Are there EEG measures that relate to things like cognitive function? And I'll show you an example of that. The other big issue, and again, at the end, we're going to talk about how we tackle some of this, is that there's a lot of variability in the methods and the way we process our signal, the way we collect our data, the way we analyze our data. All of those things drive our results. And so if you look at for instance, the coherence review, every single study used a different measure. And rather than taking kind of an unbiased data-driven approach to say, let's actually let the brain tell us where the networks are that come out, what happens instead, because of small sample sizes, is we go in with a targeted network. We say we want to look at left frontal temporal circuits, and that's what we're going to focus on for our measure. And so when you look at all those studies, they're all kind of asking slightly different questions with different variables of interest. And so that makes it very hard to replicate, but it also leads to this big body of research without a consistent message, if you will. So it's in that setting that I'm going to now give you an introduction to the kind of EEG studies that we have been doing. And again, I'm going to give you snippets of data in 3 areas, and I'm happy to answer questions about details afterwards, because each of these has in the past been, talks in their entirety. But when we think back to sort of this issue of using EEG to inform clinical trials fundamentally, what are the things that we'd like to do? So these are the 3 premises that really drive the research that we do at UCLA, and really I'd say with collaborators across the country in biomarkers. So one is that we believe that we can use EEG to study patterns of atypical brain development that precede autism and reflect a risk for autism. So that in itself, hopefully makes sense as we think about the underlying biological underpinnings, if you will, of ASD. So we should be able to measure these things really early in development. We also would propose that we can use EEG to stratify individuals and identify meaningful subgroups within the autism spectrum. And then lastly, and this is where there's, I would say, probably the most kind of proximal potential for movement, is that we can identify EEG patterns that relate specifically to genetic etiologies, which then we can leverage in trials. And so I'm going to show you examples of each of the 3 of these. But before we get into examples, what we really have to think about is being as rigorous as possible in our methods, so that they can be replicated, and so that they're really, again, taking as kind of an unbiased approach to be able to capture meaningful brain signal across a whole spectrum of patients. And so we've spent a lot of time in my lab, and I list at the bottom just some of the papers that have done this work, but there are others as well. We've done a lot of work in both the data processing piece, but first in just the data collection. So because we focus largely on very young kids and also children who have pretty significant comorbid intellectual disability and other challenges, we spend a lot of time making sure they enjoy actually coming into the testing session, which enjoy might be a stretch, but they at least tolerate it more than they would if we just brought them in and put the net on their head. So we actually have them come into the lab a few times so they can see the space. We actually have videos that they can watch. This also helps the parents who sometimes have anxiety about doing any kind of imaging or EEG studies. We have training nets that we have that we send home that the kids can wear for desensitization. This is one of our kiddos with Do15Q syndrome who loved her net so much she wore it everywhere. I actually don't think we got this one back. We just gave it to her because she loved it so much. And then we have these visual schedules that were developed by a lot of our interventionists that help, again, give kids just a nice sense of what they will be expecting. In the session itself, we actually realized that it wasn't really the child's IQ or overall functioning that was driving how well they did and what their signal even looked like. It was how kind of restless and irritable they were in the session, which is intuitive. But when we looked at the literature, we realized no one had actually found a way to quantify a child's perceived state in the testing room. And so we developed, and this paper should be out very soon, we we developed a 5-point Likert scale, very simple, called the perceived state rating, where we basically just quantified the child's state during the testing session. Now, again, our goal is not to get a whole range of states. We want kids to be happy. We want them all to be ones. But unfortunately, despite our best efforts, sometimes they're not ones. And what we found, which was really interesting, was that, and I don't have this graphic here, we found that IQ did not relate at all to amount of data retained. What we did find was that kids with a low PSR, meaning were not particularly irritable, gave us much more data than kids who had kind of that low state rating. Again, very intuitive, but nice to actually quantify to show that kids with IQs of 50 can do great in the session. They often do better because they have done tons of intervention and actually know, when it's time to sit still and engage in something, especially if then there's a nice reinforcement at the end. They're very, very, very compliant and great kids to study. It's really how they feel in the room. We also found, though, and this is really important actually, is that not only did data retention relate to this PSR, but actually EEG signal did too. And we looked specifically at alpha oscillations, which again, we care a lot about as a marker of brain development, but we also know that alpha is largely modulated by state. When we're all in a happy, restful, awake state, our alpha rhythms are very prominent. And so what we found was that the blue is typical development, and then within ASD, this is our low PSR group, our high PSR group. What we found was that alpha was lower in our kids who are more restless and agitated, had lower PSRs. So their state is actually affecting one of our main outcome measures of interest. And so we take this data and we actually account for it and control for it when we're doing our analyses. So for the processing piece, and this is going to be a lot on one slide, but I just want to put this out for those of you who do have an interest in EEG. This is our very rigorous processing pipeline that's been developed by a big team, led largely by Abby Dickinson, who's a research scientist in the lab, and then Damla Centurk, who's a phenomenal biostatistician. And we base this on reading hundreds of papers on EEG and thinking about what pipeline would we like to develop to maximize our chance for really accounting for noise in the system and generating good signal, while again appreciating the fact that we have a lot of kids who cannot sit for 30 minutes and do recordings. And so we collect data on a 128 channel system. We sample at 500 hertz. We actually manually inspect our data and we remove bad channels. And again, in the adult literature, there's a lot of automation in artifact detection and rejection. And in developmental populations, it's very hard to automate this completely. You really have to be in the data to remove some of this. We then interpolate actually down to a 25 channel montage, and we use independent component analysis to identify brain components, but also to identify the components that are clearly artifact, like eye movements and saccades and EMG, which is muscle activity. And then we remove those components so that we're left hopefully with mostly brain components. We then, and again, this is really just for those of you in the room who are really interested in this methodology, but I put this here because I think it's important to put this out up front when I show you our data. We then transform that channel level data to current source density. And this actually is a reference-free way of really looking at kind of the sources of the signal. And it also really allows us to account for the problems in volume conduction because of the fact that kids have different head sizes and hair and other things. And it actually is probably the best way to account for a lot of the noise EMG that we see. So it's a really nice pure way to focus purely on brain sources. We then calculate our power spectral density, and we can examine power in different oscillatory bands. We also measure peak alpha in a lot of our studies, and I'll show you that in a minute. And we do that using Gaussian curve fitting. And then when we examined connectivity, which is really one of our fundamental areas of interest, we use phase coherence because it seems to be the least vulnerable to volume conduction and some other challenges we see in this kind of messy data set we're working with. And we take a fully unbiased approach where we're looking at actually the frequencies across all bands in every channel. So we have 25 channels, every possible pair. You can imagine that's quite a bit. And we use this non-parametric permutation testing to ask, with all the multiple comparisons, determines which networks robustly still emerge as differentiating groups or relating to specific phenotypes. So this is a very conservative approach, but it allows us to let the data drive our findings, which I think one might argue, oh, it's less hypothesis-driven because you're not focused on a network. But the way I look at it is we are hypothesis-driven in that we believe that there should be differences in connectivity. Exactly which networks, we don't actually entirely know, even based on looking at the imaging literature and the genetics literature. And so we'd like to use this sort of big data approach to let the data tell us what we find. Okay, so in that setting, now I'm going to show you some snippets of data. So we'll start with talking about infants. So how do we study high-risk infants? How do we study autism before it even emerges? And there's several ways to do it. We could assay the general population, but that would require a study of thousands and thousands and thousands of children coming into the lab wearing EEG nets, and that would not be feasible. And when I say environmental risk, I mean things like prenatal drug exposure, other prematurity, anything else that's sort of non-genetic, if you will, although some of those kids have genetic etiologies as well. Familial risk is really what the field has leveraged. These are infants who have older siblings with autism, and these are infants who have a risk of about 20%. So about 15 to 20% of infants who have an older sibling with autism meet criteria for autism. Another 30% have kind of broader neurodevelopmental sequelae, such as global delay, language delay. And there's quite a bit of research on that. We'll talk about it in a second. And then we can also study specific genetic syndromes, which are more rare. So there's less of them, but their penetrance for autism is much higher. And we have these longitudinal study designs where we can capture these babies as early as possible. So with the siblings, we can see them at birth because we know they have an older sibling. And we can follow them closely, all the while giving feedback if we see delays so that they can get interventions. We can follow them and then look at outcomes at 24 or 36 months. And the familial risk work actually has been already been done through large collaborations that actually now, and I see the folks in the room right now, Allison and Alicia, that is now being largely supported by the Autism Science Foundation. But the BSRC, which stands for the Baby SIBS Research Consortium, is a large group of investigators around the world that are studying these babies. And what that group has been largely focused on until now has been behavior. And what they have found is that in the second year of life, we can pretty reliably identify behaviors that predict autism. And again, autism we're diagnosing reliably by age 3, but we can make diagnoses a bit earlier, but we think of 3 as sort of a good, solid benchmark for the diagnosis. And a point that was made in a recent paper from the group was that reliable behavioral markers have not been found before 12 months of age, despite the fact that hopefully I've convinced you or you already know that we should be seeing some changes in that first year of life. And so recently, and Alicia was, sorry, I know, it keeps falling. Recently, we actually repurposed the vision and mission statement a bit of the BSRC to start thinking about what is like the next phase of the BSRC. We focused on behavior, and it's been incredibly impactful to show that we can find behavioral signs of autism before a diagnosis. And our vision is to optimize neurodevelopment and well-being. And so we actually came up with 3 mission elements. And the first one I just want to emphasize is that it's focused on biology now. So we'd like to actually focus our efforts in the next few years on discovering biological mechanisms and behavioral processes that contribute to risk and resilience. And so in that setting, thank you, the UCLA infant sibling study, which we had, was funded by our first ACE, and my co-investigator here is Morella DiPreto. We focused on just imaging and EEG in the first year of life. Now, this is not work that's been published just because we're still getting outcomes, and so we're anchoring some of what I'll show you, which is kind of new data, really on 18-month outcomes based on ADOS, based on the ADOS, which is a standardized clinical measure. So we will have our formal outcomes in about a year, and we just got refunded for our next ACE, which is will continue and expand this work, and I'll show you that in a second. But we really were working under the premise that we can identify differences in connectivity and brain function in the first year of life. So this is a study integrating MRI and EEG. We start seeing the babies at 6 weeks, but the EEG is done at 3, 6, 9, and 12 months, and then outcomes at 18 and 24. Now you'll see when I show you my data slide that I wish we had EEG on later outcomes, because a lot of what we see is changes in trajectories. Okay, so here are the results. So we have thus far 26 low risk, 24 high risk, and 6 of those are showing concerns on the ADOS. And so what we've done now is look at concerns versus no concerns. And I showed this figure of the unbinned power analysis. So this is the power spectral density plot again, over all the frequencies, going all the way out to 50. And I show this because there was a recent paper that did show some differences at 3 months in power using slightly different processing techniques and a smaller sample size. It does not mean that that study was not accurate and that ours is. It just shows that, again, especially these really early ages when there's a lot of variability in these brain patterns, that we sometimes don't replicate results exactly. And so I put this out there just to sort of reinforce the point I'm going to make at the end, is that we need big consortia around this kind of work. Oops. But we don't at 3 months see differences in power in our babies. However, starting at around 9 months, we do see differences in alpha oscillations in the kids who go on to have ADOS concerns. What we do see, though, are differences in both theta and alpha phase coherence, not at 3, but at 6 months, with this picture of kind of long-range overconnectivity that quickly sort of declines, if you will, over that next 6 months. So we see this kind of overconnectivity that then really falls off. And again, wouldn't we like to have points out here? Because my suspicion is that, based on the work I'll show you in a minute, we're going to actually see underconnectivity by the time they actually are meeting criteria. So what could this be rooted in? Could it be an aberration in synaptic pruning? Absolutely. Could it be a disruption, an active disruption in white matter development and integrity? Absolutely. But we're seeing both two themes. Change is important, but that before behavioral symptoms, we do see differences in connectivity. And again, this is using that kind of unbiased permutation approach to look for robust networks. We do find that at 6 months, that coherence does relate to the social affect severity score on the ADOS at 18 months. So it does relate to symptoms in that way, which is perhaps not surprising because the difference is in kids based on concerns. So the kids with the most concerns here do show the largest difference in coherence. So the other high-risk group that we've been studying is a genetic syndrome called tuberous sclerosis, and I won't get into all the details about TSC, just to say that it's an autosomal dominant condition that is defined by having benign growths or hamartomas in different parts of the body, including the brain. It's been coined as a synaptopathy because we know that basic mechanisms of really white matter development and synapse function and formation are affected in TSC. And we've done studies for many years that have looked at the emergence of autism in TSC, and I'll mention one in a minute. But we know that the rate of ASD and tuberous sclerosis is about 50%. And what's really key, and the reason we started studying these infants, is that they're diagnosed often in utero. So this is just an example of a brain MRI showing the tubers, which you can appreciate if you're not neurologist. These white splotches are not supposed to be there. Those are tubers. But this is a cardiac ultrasound in a baby, and this little circular entity here is a cardiac rhabdomyoma, which is a benign cardiac tumor, which can get picked up on prenatal ultrasound. And so these babies are often diagnosed early. So when we first started sort of looking at these kids with TSC, we thought, well, this is another high-risk population we can study longitudinally, just like we've done with the baby SIBs. And so we've had 2 rounds of longitudinal studies. All of the behavioral studies data have already been published, and I did this work in collaboration with Chuck Nelson in Boston. This is Candace Varson, who's a fantastic now junior faculty in Perth, and then Nicole McDonald, who's a postdoc in the lab, who's also been really instrumental in this work. So we had a longitudinal study design. Bottom line is 22 of our 40 infants with TSC developed ASD. They all almost had epilepsy, and they all had cortical tubers. And that's an important point when I show you the EEG data, because despite all that cortical pathology, we can still use our approaches to generate this nice signal. So quickly, just from a behavioral standpoint, what we find in these babies is that as early as 9 months of age, the AOC, which is a very nice measure developed by Lonnie Zwagenbaum of social communication skills, the AOC is already differentiating the kids who are going to develop autism. So we actually have pretty good behavioral assays in these babies quite early on, but we can really predict autism in these babies as early as 9 and 12 months. And moreover, just to sort of alleviate any skepticism around, is it really autism? It's a genetic syndrome. What are their symptoms really like? We did a profile analysis of ADOSs in our toddlers with TSC compared to non-syndromic age-matched toddlers with autism. And what we found, this is on the ADOS, which is a standardized measure of autism diagnosis, and we found that the symptoms in TSC and non-syndromic ASD overlapped almost perfectly. So they're showing very similar social communication phenotypes to those without TSC and autism. Okay, so what about the EEG? So we saw these babies as early as 9 months, not before. And again, remarkably to me, we did not find differences in EEG power. And again, I think that's pretty surprising yet kind of nice that we, despite all of their epilepsy and everything, that just pure spectral power is kind of preserved in these babies. But again, where we find differences is in long-range alpha coherence. And we find that the babies at 9 months of age have higher coherence than the babies who don't go on to have autism. And again, we see actually a similar decline in their coherence. So meaning we see this progression from overconnectivity to underconnectivity over 2 years, from 9 months to age 2. This is the whole group, and this is just the kids that had all 3 time points. So this is kind of cross-sectional longitudinal, which is probably not quite as representative of what the change in each kid. This is just the kids that we had all 3 time points on. And so purple is the kids who actually develop ASD. So we see a real difference in the development of these cortical connections in these babies. So to summarize this portion, what we have found is that EEG power may not be all that sensitive to atypical development, but that changes in alpha band coherence may actually be a very relevant biomarker of changes in brain development. And so how can we use this? Well, we could use EEG to further stratify our risk groups before thinking about early intervention trials. We could use EEG to track response to intervention. And what we've learned here might help inform biomarker selection in older children with autism. Now you might ask, well, you're doing all these natural history studies and you know that autism, we can see signs early. What are you doing about it? What about early intervention? And I would totally agree with you, which is why in our new ACE, we actually are adding in a few features. So our new ACE, which I called Beyond Baby Siblings, is a study where we're looking at kids with familial risk compared to 2 specific genetic conditions that are highly penetrant for autism. We're again doing intensive brain imaging in the first year of life and looking at outcomes. But at 12 months, if they're showing red flags for autism, they get enrolled in an early intervention study that's run by Dr. Connie Cassery. And actually, we just received, we're 6 months in to an R01 that I now have that is actually studying the effects on both brain and behavior of early intervention in TSC. The study is called JETS, Jasper Early Intervention for TS. I love acronyms, so we make little logos for every study we have. The study is actually called Baby Bibs, but the JETS study, which is in collaboration with Chuck Nelson and Connie, we're actually studying the effects of Jasper in babies with TSC. And this is pretty profound and exciting for us, I think, because it's really the first study of a randomized clinical trial of behavioral intervention in a genetic syndrome. And so we really hope, and we're actually already seeing some, based on pilot data, some nice signal there, but we're asking whether both EEG and behavioral changes can be quantified in these babies. And I can tell you more about that afterwards if you're interested. Okay, so I have about 10 minutes, and I'm gonna use those 10 minutes to talk about these last 2 areas. Is 10 minutes right, Paul? Is that okay? Okay. So this is actually a quick one because this is a much taller order, is using EEG to stratify kids across the spectrum, right? That takes big sample sizes. And at the end, I'll talk about how we're doing that with bigger sample sizes. But we've taken a stab at this in our lab based on the pipeline that I talked about. And we've had a cohort of kids that have come through our biomarkers core as they've been a part of other studies. And so we've gotten to sort of bring them in and examine some EEG features in these children. And so we have this cohort of kids with ASD with a wide IQ range compared to typically developing kids with a less wide IQ range. And we looked specifically actually at peak alpha. And this is something that Abby Dickinson in my lab, who's again an incredible research scientist, really spearheaded because of what I told you about peak alpha being a really sensitive measure of development. We asked whether it might be a good stratifier in ASD. And in fact, we found that it was. We found overall that kids with ASD have lower peak alpha than TDs. But again, a lot of overlap. And this is the kind of scatter plot you'll see with any EEG study you find. It's not like they're completely separate, right? So there is quite a bit of overlap. But what was more notable is what we found is that as expected in the typically developing group, which is in red, we found that peak alpha related very strongly with age. But it didn't in ASD. In ASD, it related very strongly with IQ. So in ASD, peak alpha seems to be a very sensitive measure of cognitive function and development. And so that actually, again, opens the door for this to be used as a potential stratifier and maybe even a marker that might precede a change in cognition in a trial. We also took that unbiased approach, and this is in a resubmission right now, so hopefully it should be out soon. We looked at alpha coherence, like we talked about using that kind of non-parametric permutation testing approach, data-driven approach. And what we found, interestingly, and we compared TD to ASD, that same cohort, and what we found was that the networks that came out as being significantly different between the groups, using, again, this unbiased data-driven approach, were these long-range interhemispheric connections in ASD. And so this one was actually the most significantly different, where it was lower in ASD. So that as ASD as a whole, we're finding this, again, this phenomenon of underconnectivity in long-range networks, using a very unbiased approach. So we have not yet looked at how this might stratify kids. It does not relate to IQ. But this will be something that we do in ongoing and future studies. And I sort of talked about that, so we'll skip over to that. So the last area, and again, I'm giving you kind of this whirlwind tour just to give you a sense of how we kind of approach these studies. This last area, I think, again, is an area where we've been really excited about work in biomarkers, which is in autism genetics. And this is the last group that I need to be giving a lecture on autism genetics, because I think Simon's has really been the group that's, I think, spearheaded most of the insights in autism genetics. But we do know that there's many CNVs, largely de novo, as well as single gene variants and mutations that are related to and cause autism and neurodevelopmental conditions. What we know is that from a clinical standpoint, we actually can take these various CNVs and think about whether they represent meaningful syndromes. And we can also then start understanding, well, what is the genetic mechanism underlying aberrant or different changes in brain development in those specific syndromes. So it allowed us to be much more targeted in our approach. So when we think about biomarkers research in genetics, there's both challenges and opportunities. The challenges are we're dealing with really small sample sizes. The other challenge is we're dealing with populations where there is an incredibly high comorbidity of epilepsy, which makes EEG studies hard, although, as I showed you with TSC, we can still really quantify some of these basic biomarkers quite robustly. And we also, again, these children have comorbid, often intellectual disability, motor impairment, things that make them a bit harder, perhaps, to study. And it also makes it harder to apply some of our more traditional clinical outcome measures. But there's also a lot of opportunities there, and I already mentioned a couple that we can think more about targeted mechanisms and a translational potential because we have preclinical models in some of these conditions. And then I would say this is a huge one, and I'll mention this at the very end, is that many of these syndromes now have very engaged patient alliances that are pulling together resources, pulling together families to really move forward our research goals and questions, really with the goal of having clinical trials. And so that's something that we can leverage quite a bit. At UCLA, we actually started, and Paul mentioned it, this developmental neurogenetics clinic, which our parent advisory board has instructed me we have to rebrand. They hate the name. So we actually just made a new name. We thought of a new name called BAND, which is Behavior and Neurogenetics Developmental Clinic. And then we have a logo around that now. But for now, it's still DNG. What we do in this clinic is it's for any child with a suspected or known genetic etiology for their neurodevelopmental condition. They come in, they're seen by a whole array of specialists. And then after they're seen, they're actually consented into being in studies, one of which is a study funded by our IDDRC, Intellectual Disabilities Research Center, and it's my study that I've called, because I told you I like cute names, Brain and Behavior and Genetic Syndromes, where we're actually studying both EEG biomarkers and some phenotypic measures in these kids. As a quick aside, I will say one thing we do that's really, really cool, and we will be publishing this soon, is that we actually do a diagnostic journey interview with these families and ask them about the process of getting a genetic diagnosis, from the time of concerns to diagnosis, what the process was around finding a specialist, that whole process. And it's actually inspired by some work done by both Dan Geschwin and John Constantino in their ACE network. But I will just quickly say, relevant to maybe some of you in this group, and we can talk more at the end, is that some of the results were really striking. So first concerns of the parent to getting a diagnosis is 30 months. That's a very long time in a child's early development. Receiving a diagnosis and seeing a genetics specialist, and that included neurology, genetics, developmental peds, anyone who could give them guidance on the diagnosis, 13 months, so over a year. How many got genetic counseling around the diagnosis? Only 60%. So 40% of these families came to us with like a microarray report and having no idea what it meant. And then we asked, what's your main priority? What would you like to see us move forward in this field, for your child's care? They all said clinical trials. They want clinical trials for their child's condition. So I think that's really compelling, and it drives really, I think it inspires the work that we do. So within this setting, one condition that we've been studying, and again, I'm going to really just show you a couple of minutes of this as an example. I won't go through all the details in the interest of time, is Dup15q syndrome. So this is a copy number variant that's highly penetrant for global developmental delay in autism. It has some really important genes for brain development, UB3A, which is an imprinted gene, as well as several GABA receptor genes that lie in that region. But it's a complicated region because there's like 50 genes that are likely relevant. UB3A has been the one that's been most researched because when it's deleted, it can cause either Angelman or Prader-Willi syndrome. So we know that it has really important functional implications to brain development. These these children have hypotonia, global delay, autism, epilepsy. We've published some papers on the phenotype of this syndrome. What we know, and this is in a large cohort study that we've been doing, and this is just to give you a sense of what these kids with genetic conditions often look like, is that they have delays cognitively. This is the Mullen score, with a T-score of 50 being considered quote unquote normal. But we also find read as epilepsy, that the kids with epilepsy are definitely more affected than the kids without epilepsy. This is on the Vineland and the Mullen. So we're dealing with a very heterogeneous phenotype, but with children who have significant developmental delays. But we got interested in Dup15q because there was a paper that came out a few years ago that kind of somewhat anecdotally had noticed that there are these high frequency oscillations that seem to be found on clinical EEG. And they look like what we see in patients who are taking benzodiazepines or drugs that are hitting the GABA receptors. So GABA allosteric modulators like Ativan, Valium. If you give someone Ativan, this is what their EEG will look like. So that became very interesting to us because, of course, there are 3 GABA receptor genes that are overexpressed in that region, and we also know UB3A causes the co-release of GABA. So somehow is this beta signature kind of a biomarker of the underlying genetic mechanisms of this condition? And so we've been in the midst of doing a study, but I'll show you the data, some of which are already published and the bigger cohort will be. We did a study where we actually leveraged the Patient Alliance, and we collected some data at UCLA where we compared kids with Dup15q to non-syndromic ASD who were IQ matched. But then we went to 2 consecutive family conferences, and we brought our whole massive portable system, and we plonked ourselves down and we collected EEG data on a whole cohort of children with Dup15q. We also did some clinical testing and gave parents feedback on that. And what we found was, in fact, that these high beta oscillations were a biomarker of this condition. And I'll show you just the data quickly. This is a power spectral density plot again, and you can see in orange kids with Dup15q syndrome compared to the controls. And you can see very easily that these beta oscillations are much higher in this group. This is just another view of this with kind of a heat map, a topo map of the beta oscillations. And this is just showing you kind of the range. This is in our first cohort before we saw that big second cohort, higher beta across the board in Dup15q with an effect size of about 1.7, which is quite high in these kinds of signals. In our larger cohort, what we found is that we've asked about relations to phenotype. It does not seem to relate to age, so it seems very stable across time. But it does in our kids without epilepsy, because many of our kids with epilepsy were too low to even do ADOS testing, that it does have a loose correlation with ADOS severity. It has a very strong relation to epilepsy, and I'm not going to spend a lot of time on this in the interest of time, just to say that the one major predictor of beta power outside of having Dup15q, so within the Dup15q group, is the presence of epilepsy, whereby children with epilepsy have significantly lower beta oscillations than children without epilepsy. And this is just another example of that. This is a curve just showing the difference between epilepsy and non-epilepsy. So lower beta in the kids with epilepsy, but they have higher low frequency oscillations, and we think that this is a result partly of the fact that this might actually be somehow a biomarker of some sort of protection against epilepsy. Is that possible? Maybe. But also that it's actually probably reflective of the broader electrophysiological kind of environment in our patients with epilepsy, where there may be more of a contribution of these lower frequency oscillations that might kind of cloud some of those beta frequencies. But regardless, it's very robustly related to epilepsy, which is important when we think about designing our studies. And I'm almost done. So we've then asked, the question about scalability. We know we can measure this really easily. could we actually quantify this in clinical EEG? So we just got a grant to look at clinical EEGs in kids with DUP15Q around the country. And what we find so far in our first cohort is that we can see, just like we do with our high density recording, we can quantify these beta oscillations on their clinical EEG. So that opens the door a lot to do this kind of work more robustly in the community rather than having to have them come into the lab and wear our net. And this just shows the stability between the two recordings. And so to summarize this part, I'll say that what we found in this one condition is that beta oscillations relate to DUP15Q syndrome. So imagine taking this through kind of a pipeline where we might diagnose a child with hypotonia, autism, epilepsy, and with genetic testing we find they have DUP15Q syndrome. We then actually identify these high beta oscillations. Well, imagine if we have a medication that targets, for instance, these GABA receptors through either a GABA antagonist or any kind of drug that might modulate the system. We might be able to use those beta oscillations as a marker of target engagement of the drug. It might also be a proximal outcome measure in itself in a trial. And so, and in perfect timing, I'm done. Okay, so we know that EEG, I'm going to just end by saying that EEG is a really promising tool to study brain-based biomarkers in autism. I'd say they're grounded in mechanism, they're developmentally informed, and they're practical and scalable. But my big kind of word of caution is that I think we're still really constrained by small sample sizes, variability in methods, variability in methods. And so I think we need to move towards not just consistency, but collaboration. We need an infant biomarkers consortium because otherwise we're going to continue to have, I think, these small studies that are difficult to replicate. I think we also need some rare disease consortia, just like we've done with DUP15Q, across conditions and within conditions. We need mechanisms for pooling clinical EEG data so we can think about whether some of these biomarkers, not just the beta oscillations, but some of these other ones I talked about, could be actually pulled from clinical recordings, because that would actually open the door quite a bit in terms of scalability. And then we need common data platforms and mechanisms to pool multimodal data. I think this is really key, and I know Simons and others have been really involved with thinking about this. I won't go into detail here, but I'll say there are some efforts in this already. We have a biomarkers consortium study called the ABCCT that's targeting 6 to 11-year-olds with autism, funded by NIH and Simons as well, that's really asking this question about biomarkers across sites, and UCLA is one of the sites for that. We also have this group called Agenda that Alicia Halliday here has been really instrumental in. And this is an alliance of patient advocacy groups with researchers and clinicians in rare diseases to think about these questions that I've just asked. Sort of, how do we pull data? How do we develop patient registries that help us move forward in clinical trials? We had our first patient registries meeting that was very nicely funded by Simons. And we also have a special interest group at INSAR. We had our first one last year. We have another one coming up this year. So we are moving in this direction of big data and collaboration, but I would say that we really are just at the beginning of this, and we need to kind of continue these efforts. And with that, I'll stop and just say thank you so much for inviting me. This is my lab, although we've grown since that picture. I sort of showed pictures of the folks who've done the work along the way, but really appreciated. Our funding sources are largely the NIH as well as the Department of Defense and the DUP15Q Alliance. So thank you very much.